Greetings, Brookings AP Bio students. This is Mrs. Rydell, and we're going to continue with our slideshow packet of looking at Chapter 17. How do we get the information that's stored in our DNA code, turn it into an RNA message, and pass that RNA code out to our cytoplasm where it's going to be read by the ribosomes and make our protein. In our last little video that you watched, we looked at the first part of this process, going from DNA to RNA message. We looked at transcription. In this little video, we are going to look at taking that message now, turning it into a protein. Ribosomes out in the cytoplasm are going to do that. They're going to read the message, and they're going to build a protein. And the protein now, remember, is going to go on to show itself in the cell. And that process of going from RNA message to protein is called translation. We're going to look at that process of translation and how we take the code now and pass it out to the cell. And ultimately, proteins are the way that genes are going to be expressed. Some sort of a trait, some sort of phenotype in the cell is going to show. So if you look now at that process, we're going to be switching language. When we did DNA to RNA, we were still talking the same language. We're talking nucleotide language. We use nucleotides to build DNA. We use nucleotides to build RNA. But now when I go from RNA into a protein, I'm actually going to switch language. I'm going to use a different alphabet. I'm going to use, instead of nucleotides, I'm going to change my nucleotide message into an amino acid message. And that's why it's called translation. We're switching languages. So how does RNA code for proteins? If you remember, we learned about this back in biology. So this should be kind of a review for you. Remember, DNA exists as a sequence of 4 different nucleotide subunits, A's, T's, C's, and G's. Those nitrogen bases on those nucleotides make the code. And now we're going to take that DNA information in the DNA and turn it into an RNA message. We're still using nucleotides. Remember, our choices are a little different, A's and U's and C's and G's, but we're still talking nucleotide language. And we have 4 choices for those nitrogen bases in my RNA message. Now we're going to switch message. Proteins are made up of amino acid subunits, and so 20 different amino acids are going to be used to build proteins. And so what we have to figure out now is, how am I going to go from a 4 code, 4 letters in my code, to make 20 different amino acids? And the way we do that is we're going to read it in little groups. We're going to make words out of the DNA code. So if you look at the information now, we're going to read it in little groups of 3s. So each little group of 3 is going to stand for 1 amino acid. Each amino acid has a different codon. And so that's how we're going to be able to take a 4-letter language and make words. The little groups of 3s that are in the RNA message are called codons. They're carrying the information. And so how did we figure out the code? Actually, Crick from Watson and Crick fame was the guy who figured out that had to be a 3-letter triplet system. And then 2 other scientists, Nuremberg and Corona, working in different labs actually, worked out the what the codes stood for, determined the mRNA amino acid match. What was the mRNA code that stood for each amino acid? They did that by making artificial mRNAs in test tubes and combining them with ribosomes and tRNA and amino acids, and making artificial messages, and then looking to see what amino acids were put together from reading that message. The very first one that was figured out was phenylalanine, and the code for phenylalanine is UUU. And so they created an artificial mRNA message by hooking together uracil, little subunits, and turned out that the ribosome read that and made a chain of phenylalanine. So they knew that the very first code, the UUU code, stood for phenylalanine. These guys are the ones that got the Nobel Prize for figuring out the code, and that was 1960s. And we can use that code now to figure out what kind of amino acids are going to go in for different codons. That universal code is shared by all living things. So it's kind of evidence for the idea that living things are connected, and that idea of shared origin, because we are all speaking the same genetic language. In bio, when we did use codons to determine sequence, we used a kind of a wheel type decoder wheel, but we're going to use a little different decoder this time. If you look at how we read this, the first base in the code, remember we're reading in groups of 3, are listed along the side here. So all of these in this row have G as their first base. All of these in this row have C as their first. Okay? And then second one is listed across the top. So all of the ones in this column have U as their second base. All of the ones in this column have G as their second base. And then as I go across and read the code, the third base in our code is in the last, shown along the right-hand side here. So if I have a code that says, for example, U for first letter, U for second letter, and then A for my third letter, it's going to tell me leucine is my amino acid. Notice the code is redundant. There's multiple codes that code for the same amino acid. Proline, there's 4 codes that code for proline. CCU, CCC, CCA, CCG. Some have even more than that. Look at leucine. Leucine has 6 codes that code for that amino acid. Arginine is another one. There's 4 here and 2 here. Arginine has 6 codes. Some of them only have 2 codes. But the code is redundant. There's more than one codon that stands for each amino acid. And notice there's a little wobble, what we call wobble, in the third base. For all these codons, the first 2 letters are the same. And the third letter could switch. We could have a mutation in that third letter, and it's still going to put in the same amino acid. So that's the wobble, that third base wobble. It allows the cell to have a little mutation there, a little change in that last code letter, without actually changing the protein sequence very much. We also have some codons that code for start and stop. So AUG is our start codon. It's going to put in the amino acid methionine. That means this is the starting sequence of every protein. Every protein is going to start with methionine and that first, as the first amino acid in the chain. We also have some stop codons. 3 stop codons, UAA, UAG, and UGA. And those, when the ribosome gets to those codons, it's the signal that says stop, you're at the end of the line, we're going to stop adding amino acids into the sequence. So think about the little code now. You do not have to memorize all these codes, but you should be able to read a code wheel, and if you were given a sequence, be able to determine what the amino acid sequence would be from those codes that you are given. So think about the wobble. Why do we have a wobble? It allows us to have a little change in the code without impacting the protein. We can absorb some of those differences. So how are codons matched to the amino acids? If you remember now, we're going to use the code little sequence in the message and reading in little groups of 3s. And the amino acids are going to be brought in by carriers, transfer RNAs. They're going to transfer in the correct amino acid. And how do we know what place to stick it? They're going to match a place on a code on the tRNA that matches with the codon on the message. And it's going to bring in the correct amino acid and put it in the right spot. That sequence on the tRNA is called the anticodon. Anticodon is going to be complementary to the codon. So GUA is going to match CAU. Remember, no T's. This is RNA now. So we're going to match up the codon and the anticodon, and that tells us we're in the right position, and that tRNA is going to bring in the amino acid and put it in the right spot. So we're going from code DNA in the nucleus, we're going to make RNA, that's transcription, and now RNA is going to be turned into a protein by the ribosomes. And tRNA, the tRNAs are the delivery trucks. They're bringing in the building blocks to build the proteins. So my protein factory is going to receive the amino acids and hook them together to make a polypeptide chain. That process is called translation. And protein then goes on to make the trait. Blue eyes, height, intelligence, skin color, hair color. Makes an organism. Okay, so let's look a little closer at some of the RNAs that are going to help this process of protein synthesis. First of all, transfer RNA has a very unique clover leaf shape, and that is the RNA that's going to be bringing in the correct amino acid. Notice it kind of has this clover leaf pattern, anticodon here, kind of in the middle, and this end, my 3' end, is going to where we attach the amino acid. Notice we have some double-stranded 3D structure going here. This little piece of tRNA is actually going to fold up into a 3D shape, and the anticodon is going to be at one end, and the amino acid is going to attach at the 3' end. And notice there are some little different ways to represent tRNAs. This is kind of the sequence. We can see it all stretched out. This is when it's folded into its little 3D shape, and then we can kind of do this way to represent the overall structure with amino acid at one end and the anticodon at the other. So how do we get the amino acids onto the tRNAs? There are enzymes that do this. Amino acyl tRNA synthetases are the enzymes that are going to add the amino acids onto the tRNAs. This bond, making the bond to hook those on, requires energy. And notice now we're going to take the amino acid and attach it to the end of the tRNA molecule. And this is look what's going on. We're removing the water. This is a dehydration synthesis reaction. We're attaching the amino acid to the end of the tRNAs, and then that bond is going to be broken when we release the amino acid at the ribosome. So ATP is going to provide the power to add the amino acids onto the tRNAs, and the enzyme that's going to make that happen is amino acyl tRNA synthetase. So we have a leucyl tRNA synthetase that adds leucines. We have an alanine tRNA synthetase that adds alanines. We have a phenylalanine tRNA synthetase that adds phenylalanine. Each of these has a specific enzyme, a specific tRNA synthetase enzyme that's going to add that specific amino acid onto the correct, remember, has to make it onto the correct tRNA that has the anticodon for that amino acid. So once we add, this is going to be a transporter. We're going to add the amino acids onto the end of the tRNAs, and then they're going to carry that to the ribosomes, where they're going to release them and to build the protein chain. So ribosomes now are going to facilitate the coupling of matching up the tRNA anticodon with the codon. And so if you think about it, we kind of talked about ribosomes before as organelles. They're protein synthesis factories, right? We're making proteins. But really, they're also acting as enzymes to make this chemical reaction happen. If you look now, our structure of our ribosome, we have ribosomal subunits, RNAs, and proteins that go together to make the ribosome. There's two subunits, a large and a small, that go together. And remember, we talked about sizes of subunits in prokaryotes versus eukaryotes. Eukaryotic ribosomes are a little bit bigger. Subunits are a little bit bigger than those in prokaryotes. So we're going to watch a little video of this reaction happening.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
If you look at this little video now, this video was showing you protein synthesis in a prokaryote, in a bacterial cell. The process works the same way as eukaryotes, but there are a couple differences. Notice the ribosomal subunits are a little bit smaller. Also, that first amino acid that comes in way back at the beginning of our sequence is a modified form of methionine. Remember, we said methionine is coded for by that first start codon. In bacteria, the methionine has a formal group attached to it. It's called formal methionine, but it's going to work the same way. We're bringing in the amino acids, we're going to match up the anticodon with the codon and put the amino acids in the correct position. We're going to go back to our slideshow a little bit and look a little closer at how this is going to work now. The three sites that are going to provide positions for the tRNAs to move along the ribosome are identified as A, P, and E. A for the video said acceptor site, also called the amino acid site. That is the site where the incoming amino acid is going to add and sit down in this spot. The P site, the peptidyl site, also called the polypeptide site, is the place where it's going to hold the growing polypeptide chain. And then E is for exit. We're going to empty the site. We're going to leave the tRNA once it drops off its amino acid is going to leave via that exit site. So we're going to build a polypeptide. Initiation is putting together the mRNA message, the ribosomal subunits, and that very first start tRNA that has the methionine or formylmethionine. That's the very first initiation step. Elongation is going to happen now once we get the ribosome set up with the message there. The elongation is going to happen as we bring in the amino acids and add to the chain. And then eventually we reach the stop codon that says this is the end, and we terminate the sequence. The complex falls apart, ribosome dissociates from the message, and we have our little polypeptide chain. So we're going to watch another little movie here that shows how this is going to happen.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay, so let's go back to our slideshow. And once the protein is made now, let's talk about what's going to happen to this. Remember, the ribosome is reading the message, running down the line, and making this protein chain. There is a little signal molecule that is at the end of that growing chain that is going to be recognized by signal recognition particles and tell the cell where to send the protein that's being made. If it's going to be secreted, for example, and sent out of the cell, it is going to be, the ribosomes are going to attach to the rough ER and insert the growing protein chain into the lumen of the rough ER and send that protein now where it needs to go inside the cell. Maybe going to be packaged, sent to a Golgi body to be packaged to be sent out of the cell. It may be a protein that's going to become part of a cell membrane. It may be a protein that is going to be used in the cytoplasm. In that case, then it would not be put into the ER, but maybe stay outside. And those little signal molecules at the end of the chain are kind of like stamping address labels onto the protein and telling the cell where do we send it? Where do we send it? Okay, so let's look at the sequence now. Remember, we're going to start with our DNA in the nucleus. Right? DNA in the nucleus. Our DNA is going to be read by RNA polymerase and turned into an RNA message. That message is going to be edited in the nucleus. The pre-RNA is going to be edited with introns removed by the spliceosomes. We're going to add our caps to the final message. The GTP 5' GTP cap and the poly A tail are going to be added. This helps get the message out of the nucleus and protect it from being degraded, so that the ribosomes can work on reading the message and use it to make a protein. Our mature RNA now is going out to the cytoplasm. The ribosomal subunits are going to attach onto the RNA message, and we're going to build the protein by bringing in the tRNAs and placing them in the right position. Our enzyme that's going to add the correct amino acids on to charge up our tRNAs are our amino acyl tRNA synthetases. And here's our tRNAs and our little amino acids that are going to be added to the end. We're bringing it in the A site, the acceptor site. We're putting the amino acid carrying tRNA here. The P site is going to hold our growing peptide chain, and then E is our exit site. Once we drop off our amino acids, the empty tRNAs are going to leave and go back out to the cytoplasm where they can be charged up again for the next go around. We're reading 5' to 3'. Ribosome starts reading on the 5' end of the message, and we are making a protein. Okay, let's throw some of the labels on here. Amino acids now are being charged up onto the tRNAs. Our amino acyl tRNA synthetase is going to do that, and then we're bringing in those charged up tRNAs to put the amino acids in the correct spot. We're making a polypeptide. That's translation. Okay, so let's look a little bit at what is a transcriptional unit. What is a gene? We kind of have changed our idea about what genes are, because if you think about the piece of DNA that is going to code for that message, right? Code for the message is what we would call the gene code. There are also regions next to the gene, the code that actually has the information of how to build a protein, that are regulatory sequences. Promoter sequences give a place for the RNA polymerase to attach, and we talked about TATA boxes in APBIO. That sequence of multiple A's and T's over and over again in the code help the promoter attract the RNA polymerase and make it more likely, easy to open the code. Remember, A's bond with T's and T's bond with A's with only 2 hydrogen bonds. G's and C's are attached to each other with 3 hydrogen bonds. So if you have a long sequence with multiple A's and T's over and over again, that hydrogen bonding there is going to be a little bit weaker and easier to open up the DNA and get the information started. We also have enhancer regions that are a little farther down the line upstream from where the gene is, that also help by folding the DNA and getting it into position to help the polymerase attach. And then RNA polymerase now is going to attach at that promoter spot, start reading the gene, and move down the line. There's a start signal, there's a stop signal that says this is the stop, this is the end of the gene, and RNA polymerase then is going to move along, read that information, and turn it into a message. So introns, remember, are the pieces that are what we call the junk DNA, that pieces that are not going to end up in our final message in our edited version. The exons are the pieces that have the information to make the protein. Those are going to be spliced together and make a transcript. Okay, we have starting sequences that tell the ribosome where to start, and we also have ending sequences that are going to tell the ribosome where to end. Translation stops. So there are a number of coding regions that don't actually code for proteins, that are regulatory, that are going to help get the RNA in position, that are going to tell it when to start, when to stop. And those are also part of that unit on the DNA. We're making a pre-mRNA that needs to be edited. We're going to remove the introns and take that mature message now. We're going to add our cap on a 5' end, add GTP to protect that tip. We're going to add poly A's, many A repeats onto our 3' end. That's our poly A tail. And that message now is going to go out to the cytoplasm and be translated into a protein. We are going to stop right here. We have one last little bit to talk about, which has to do with comparing protein synthesis in prokaryotes and eukaryotes, and that is coming in our next installment. So stop, take a break, and come back later. We are going to stop right here. We have one last little bit to talk about, which has to do with comparing protein synthesis in prokaryotes and eukaryotes, and that is coming in our next installment. So stop, take a break, and come back later.